on the test. So it's almost like a review. So chapter 4 is in your textbook going to be including, I think everything's at your, where you sit. And so it's going to be continuing talking about the cell, but the textbook is going to contain a ton of information. I have chosen the information that I want you to know. All right? So you'll notice chapter 4, we only have 13 slides. All right? So it's a very condensed, because it's basically, I want you to know what happens with the ribosome and how we make a protein. Because remember, proteins, they are carrying out the functions of the DNA. So that is, and of course we'll look at mitosis because we're dealing with our animal cells, and meiosis will come back to us, well, it'll come back in part 2 when you get to the reproductive system. So, if you remember, we kind of had this picture a little bit in the last chapter because we were looking at the nucleus, the nucleolus. We kind of showed that we have our DNA in the nucleus, messenger RNA, that kind of went to the endoplasmic reticulum. Does everybody remember that? If we were making a protein to go outside the cell, and that went to the Golgi apparatus, all right? And then that had to move to the membrane to either kind of like get packaged out and go out, or get to the membrane and have that product spit out. Does everybody remember that? You need to know that for the test, okay? That's why I said this is going to be a whole lot of review for the test. This time, what this chapter is showing us, this chapter is showing how a gene on our chromosome is going to help the cell make the protein. It is ribosome's job to carry out protein synthesis. You need to know that for the text. Okay? So, the way that this is going to happen, the nucleus of the cell, surrounded by this nuclear envelope, hint, hint, okay, anything I'm going to be stressing right now, you need to know that's going to be on your test, okay? So, this nuclear envelope contains these little pores. They're called nuclear pores. And inside of that nucleus, this is where we find our DNA. Do you happen to remember how many molecules of DNA are in each nucleus? Because one molecule of DNA represents a chromosome, 46. So we have 46 chromosomes, and of that, what this means, if I have 46 chromosomes, all right, it gives me 23 pairs. So, one molecule of DNA is a chromosome. On that structure of the chromosome, And don't forget, it's double-stranded, okay? DNA is double-stranded. It's held together by hydrogen bonds. And it has bases. Do you remember the bases for DNA? Tori? We have A to T. Whoa. There we go. A to T, G to C. All right? So that means along this DNA, where I might have an A, it has to pair on the other strand to a T. A G needs to pair to a C. Does that make sense? Okay, so let's just do a couple of bases. All right. All right, so if my DNA is found in the nucleus and along this strand of DNA, all right, I find genes. All right, is everybody following me? That gene codes for something. All right? So, if you remember, and like I said, a lot of this is review for your test. In the cell, we had two types of ribosomes. We had free. Does anyone remember what happens to the protein made by the free ribosome?
I mean, no, that's the one that's made in the cell that be attached to
The free ribosomes. So if I'm in my cell, I have my nucleus that has my DNA, has my endoplasmic reticulum, and then I have ribosomes out here in the cytoplasm. I'm giving y'all a bunch of stuff for your test. Okay? So, if these stay free, that protein that gets made stays inside the cell. If the ribosome attaches to my endoplasmic reticulum, it gets sent to my Golgi apparatus, and it gets packaged into a little vesicle from a protein, and that makes its way outside the cell. So this is a free ribosome. This one is attached. The ribosomes have two parts. They're called subunits. One is termed the large, one is termed the small. So when a gene, now remember, this is representing my DNA because it's double-stranded. All right? And in the case of the cell needing to make a protein, it needs to open that part of the DNA strand. And messenger RNA will copy it. Okay? So this stays inside my nucleus. If the messenger RNA gets formed, All right? That is now single-stranded. So it's single-stranded, and it gets to leave the nucleus. When it leaves the nucleus, it makes its way, it's kind of some debate, but either way, ribosomes attach. Is everybody following me? So if a ribosome attaches with the large and the small subunits, this allows for transfer RNA to begin coming in, and we begin to string together amino acids. Amino acids are the building blocks of proteins. So let's kind of take a peek at what they're trying to show us in this picture. What they're trying to show us is we have our nucleus. We've got our double-stranded DNA. It's open at this point. Note how they're trying to show us it's not open here, it's not open there. The part that will code for the protein is right here. So that section of the DNA opened, messenger RNA pairs to it. Do you happen to remember the bases in RNA? Kevin?
It's the same thing, but thymine becomes uranium.
It's now a U rather than T. So it would base pair, and that strand exits through a pore. So that single strand that now contains A's, U's, G's, and C's makes its way out into the cytoplasm. So our DNA has the information to code for that protein. The process of the DNA strand's ability for messenger RNA to make its strand, this is termed transcription. Transcription, using the DNA strand, makes the messenger RNA. And that messenger RNA gets to leave the nucleus. At this point, when the messenger RNA leaves the nucleus, that strand now contains information on how to make the protein. For example, if I drew a circle around this representing my plasma membrane, let's say that that messenger RNA strand was coding for the cell to make a channel protein. All right, is everybody with me? Do you remember a channel protein? Where are they found? In the plasma membrane. So if it was coding to make a channel protein, would it attach, in other words, that ribosome that will attach to that messenger RNA strand, would it need to dock on my endoplasmic reticulum? No, because it's making a protein that stays inside the cell. If that messenger RNA strand was coding for insulin, where would that ribosome need to go now?